Good evening, everyone. My name is Ellen Fred, and I serve as Assistant Director of Online Engagement for the University of Illinois Alumni Association. I'd like to thank you all for joining us for tonight's Alumni College webinar. We very much appreciate your support of our ongoing speaker series, and we're excited to welcome back several repeat attendees. Before I introduce our speaker for this evening, I'd like to take a moment to talk about some offerings from the UIAA that you can access from wherever you call home. First, if you're not already signed up, we invite you to join our online networking and mentorship platform, Alumni Link. You can visit alumnilink.illinois.edu to set up your profile and connect with thousands of fellow alumni from around the world. You can also join our online book club by visiting pbc.guru/uillinois. Joining the book club is free and provides our alumni with an opportunity to connect with one another over shared interests in literature, history, and professional development. We'd like to invite you to join us for our next speaker series webinar on June 11th from 6 to 7 p.m. Central Time, when we'll be joined by Chancellor Isbell. This conversation will provide an opportunity to hear updates from campus leadership and learn more about the exciting work happening across the University of Illinois community. Registration information will be shared in May, so please keep an eye on your inbox for more details. We're celebrating an incredible year for Illinois athletics, with our men's basketball team heading to the Final Four and our women's team delivering an outstanding season and tournament run. New annual and life Alumni Association members will receive an Illini Watch Party fan pack, so you can keep that game day energy going. For more information on becoming a member, please visit uiaa.org/membership. Finally, I'd like to remind everyone to please submit questions during the presentation via the Q&A feature at the bottom of your screen. It may be under the More option. Your questions will be addressed throughout the session or at the end, and we will get through as many as possible within the allotted time. If you are interested in reviewing this presentation at a later date, a recording will be posted to the speaker series section of our website within the next 10 business days. Now, let's get to the reason we've all logged on tonight. I'm honored to introduce our speaker for this evening, Rohit Bhargava. Dr. Bhargava serves as the Philip and Ann Sharp Director of the Cancer Center at Illinois, as a professor of bioengineering and the Granger Distinguished Chair in Engineering at the University of Illinois Champaign-Urbana. With that, it is my pleasure to turn the program over to Dr. Bhargava.
Well, thank you so much, Ellen. I really appreciate the chance to share some of our work with our alumni, and I thank the UIAA as well. So without further ado, I'll start talking a little bit about a very fundamental concept that I think is near and dear to many of our hearts, which is the search for breakthroughs in cancer research and cancer care. And I hope today I'll be able to share with you some of the things we're doing here on campus, how that's different, and how we're hoping to make a difference in the national scene as far as cancer health is concerned. So we have formed the Cancer Center at Illinois here. We started about 15 years ago from just a concept paper, and today we are one of the institutes on our campus. We have a very clear mission to advance fundamental engineering and basic science discoveries, and then use those discoveries to transform cancer research, detection, and treatment. Our vision is that there isn't a place in the country where you can go to and take your technological needs in cancer research or care and have those addressed. We hope to fill that niche. We want to become the national hub for engineering and basic science in cancer and use those advances to change how we think about cancer and how we treat it. So our approach is to use an engineering mindset that effectively creates, based on requirements, a solution, then looks at the solution and improves it continuously over time, personalizing it with every iteration and making sure the solutions reach every single person. I'm quite pleased to share with you that we are on the trajectory to become just the 8th National Cancer Institute designated basic cancer center in the country. And when we're designated, we will be the first new one since 1987. So it's not just us putting forth a unique kind of cancer center on this campus. It's actually a momentous event for the whole national cancer community to invest in an area that focuses on basic engineering and science to provide new solutions. The Cancer Center itself sits within this university ecosystem, and I don't have to tell you what a wonderful university we have, but what we are organized around are major global concerns that are going to be problems in the coming years. The Cancer Center is one of these 9, And this really adds to the collaborative culture on our campus so that we can all work together and make sure that the benefits of our research in any one unit reaches every other unit. We're also very fortunate to have an amazing number of faculty who are well recognized across the country. So here's just a selection of many of our faculty who've received recent honors, and you'll see many familiar names here. I'll talk about a lot of their work as we go along. But what I want to emphasize here is that the Cancer Center at Illinois is actually a pretty large organization. We have 81 what we call full members. These are people who have active funding, federal funding to do cancer research and publish papers or make innovations and get patents rather frequently. Then we have 50 associate members. These are also faculty on our campus, but they're either turning their attention to cancer or on their way to collaborating with somebody interested in making a change in cancer. So about 130 faculty members. If we were one department, that would make us one of the largest departments on our campus. But what we are is a spread over campus unit. So we draw faculty from more than 20 departments and 6 colleges. But what we try to do is bring their efforts commonly together to address this problem. We also organize in a different way. We organize in a way to take our efforts from basic discovery to developing solutions and then to translate them. Another way to think of the organization is how we organize into topics of scientific interest. So one side of the Cancer Center is more focused on technology. Here we have 3 big directions. One is cancer imaging, because you want to see what is there. You want to also understand what it is composed of in terms of molecular measurement. And cancer is so complex that we can't do this just with our eyes and on a piece of paper. So we use computational engineering and data science, including very advanced AI approaches. The other half of the Cancer Center focuses on understanding how cancer initiates and progresses, using very unique approaches to biology that I'll show you in a bit. Once we understand the biology a little bit, we also try to devise therapeutics and very new kinds of chemical compounds in new ways by fundamentally advancing the science of chemistry. Then there's something really unique on our campus, actually 2 things. One of them is our very strong material science and engineering program. We engineer tumor models in the lab, and I'll show you several examples of that. The second is our wonderful veterinary school. So we use pet cats and dogs as models for cancer because they're our pets. They encounter the same environment, the same food oftentimes, and the same risk factors that we do. And of course, we want to make sure that we take care of them. So we try to bring drugs first to our pet cats and dogs and then move them to humans very quickly. In addition to these research efforts, we also have a very active educational effort, especially in the emerging areas of science and technology that would benefit the country. So we've established over the last 15 years efforts all the way from high school to undergraduate program, graduate, and even faculty programs that allow our faculty to build successful careers in cancer research. In particular, we're being increasingly recognized across the country for leading the area of cancer engineering. And the way we think about it is it's a convergence of many different disciplines, all organized to address the problems of cancer and make sure we have better lives with and after cancer. So now I'll share with you some examples of the kind of science that is happening. And I want to emphasize 2 things. One, cancer science, cancer research, and impactful cancer solutions is a team sport. So we're highly collaborative. You'll see many people working together. Second, it's a long and systematic process. So in every example that I share with you, we'll talk a little bit about basic discovery. We'll talk about how we take those basic discoveries in the Cancer Center, turn them into impactful technologies, and then we'll talk about how they're actually being applied and perhaps the impact of those technologies that we're already starting to see. So our first example comes from a group of faculty in electrical engineering and computer science. What they're interested in doing is doing some very fundamental studies about how vision systems work. They want to go to nature and sense how do insects, for example, sense their environment. How do shrimp sense their environment undersea and so on? By this fundamental understanding, they want to design better cameras. And they rely on the wonderful facilities we have here at the University of Illinois to be able to fabricate these very unique cameras that are able to see not just in color, for example, that we are all used to seeing, but in terms of how light is polarized. So if you look at this image at the bottom, you can see sort of on the left, the color image that we would normally see with our eyes. And on the right, you would see what some insects in nature see as polarization. Now, polarization in the tissue where there's cancer changes. So by just using this camera, we'll be able to see where the cancer is very, very quickly. And so our colleagues have taken this technology into the operating theater, and now we're integrating these into goggles. So imagine a pair of glasses that a surgeon puts on, and they can see both the color as well as the presence of cancer within the tissue itself. Then we use light to sort of project where the tumor is. And as you can see in this image, they can precisely identify where the cancer is and surgically remove the cancer. So this is technology that's already making its way into operating theaters and already improving surgery in a new paradigm. My second example comes from our colleagues in bioengineering and electrical engineering, Brian Cunningham, Xing Wang, and Andrew Smith. This group is interested in making very sensitive molecular measurements. So just through a drop of blood, if there is a single molecule that is cancer responsive, they want to find it. And it all starts from this very fundamental physics-based modeling. Of course, once they achieve that, then they design instruments that are actually practical to use, inexpensive enough to deploy, and can be used anywhere in the world. So here's a schematic of the design. This is, of course, Illinois engineering at its best. So we're engineering the system to be short, compact, being able to be put into a primary care physician's office. And the idea is that if a patient comes in, we take their saliva, for example, any bodily fluid like blood, put it on a small cartridge, put the cartridge into this white instrument that you see here, and let that instrument in about 30 minutes will give you a sense of whether the markers for cancer are present or not. So we'll be able to screen for any cancer in a short period of time routinely. And of course, early detection is one of the keys to successful cure in cancer. So these kinds of devices could be revolutionary in how we address cancer, how quickly we detect it, and how we treat them once cancer is detected. My third example is from a really fundamental study, and this has been led by Zan Luthe Schulten, shown on the left, includes me and 5 other colleagues from our campus. And the idea here is, can we build a computer model of cancer? So before we can build a model of the whole cancer, we actually have to build a model of the cell. So Zan brings in the expertise to build a computer model of the cell, and my own lab brings in the expertise to record in a cell every place, every molecule, and catalog it. Then Zan takes that information and puts it into a computer model. So here we're just showing the DNA and the helper molecules that go along with the DNA. And what you're seeing is a cell divide. So there's green DNA from the original cell, and then there's the cell dividing into 2 different cells. One DNA is shown in purple, the other one shown in green, just for our visualization convenience. This is the first example of a whole cell dividing in a computer that is completely determined by physics and chemistry. So this is quite a momentous occasion, quite a momentous achievement for us as a scientific community. But again, our goal here is to study this in even more detail by putting in more complexity. So what we've done is organized a consortium, both in the country as well as across the world, brought together industry like NVIDIA, for example, brought together the leading universities, and we're trying to solve this problem together by combining the latest technology. For example, the one shown here. On the left is an image that we used to see till a few years ago. On the right is an image that pinpoints exactly the individual proteins that are labeled in this particular case that we can actually detect. So this whole consortium is going to make measurements of various constituents in the cell and feed them to Zan, who'll put them in a computer model, and we'll be able to see how that model predicts cancer and so on. So we call this bringing computational cells to life. This was such a revolutionary concept that the National Science Foundation gave us funding to actually have a center, one of only 4 in the country that's actually engaged in this high level of science to be able to make this a reality. The chemists, Zan Schulten is, actually are doing absolutely groundbreaking work. This is from the work of Marty Burke and Human Zhao. They are using artificial intelligence to design ways in which we can make new molecules. And not just make 1 or 2 or 3 molecules, but make thousands of molecules. And the idea is to make thousands of molecules that have the potential to address cancer. and then choose the best one from them and iterate this very, very quickly. So the idea here is to synthesize those molecules, put it into a computer model, see how it addresses the molecule of interest in cancer, and then move it into practice by perhaps coming up with high throughput testing and using those drugs to potentially test in humans. This is a very uniquely Illinois approach where we're not just using AI to understand how drugs and proteins interact, but we're using artificial intelligence to design the very molecules that could be the revolutionary drugs of tomorrow. Of course, this builds on a huge legacy of wonderful chemistry at the University of Illinois. Here, Paul Hergen-Rother's lab is developing libraries of small molecules. So these are fairly innocuous molecules, but once they encounter a cancer, they become very, very effective. And by screening these molecules with Cancer Center resources, they're able to select some of them to be applied to our pet cats and dogs. So here I'm showing Tim Fan with a patient who's actually treated with a drug that's developed right here on our campus. And once we can show that these drugs are helpful to our pets who have no other alternative at this point, then we take them to humans. So we've packaged many of these drugs that are suitable for humans, and there are phase 1 and 2 and 3 trials ongoing from several of these drugs. So we hope that this uniquely Illinois approach of combining artificial intelligence, high throughput chemistry, quick testing, transitioning it to animal models, and then eventually to humans would be the way of the future. But we are obviously well far down this path, and hopefully there'll be a lot of impact of this approach coming forward. So I hope you got a sense of how we are approaching the cancer problem. We call this paradigm discovery to use. We're really going to focus on basic, fundamental, foundational discoveries in engineering, in basic science, but we're also going to keep our eye forever on how these discoveries can be used to benefit those with cancer and afterwards. So our goal is to provide ever-improving quality at scale, at low cost, and hopefully personalized to people. So I gave you several examples at a high level. I'd love to share now with you 3 deep dives from my own laboratory. So these are 3 you can think of as case studies, where we'll go through and understand how basic science followed by engineering is leading to new solutions that can potentially prove useful. The first deep dive that we're going to undertake today is to ask the question, can we devise technology to provide a way to diagnose cancer better? For example, can we make it more accurate? Can we make it more immediate? Can we make it low cost? And can we actually integrate new technology where we're treating the patient rather than have this lab-based delayed healthcare system that we have today? So to understand this technology, maybe let's go back about 130 years. So around 130 years ago, the standard of practice used to be, we take a piece of the tumor out of the human body, we prepare a slide, we do optical microscopy, and then a pathologist looks at it to make a diagnosis. Unfortunately, it's the exact same process today that we follow, unchanged for more than 130 years. And here's what that image looks like. So under a microscope, you can see this image where there might be a little foci of cancer here, and a trained person who has a trained eye, went to medical school, and was trained afterwards can actually look at this. What we're trying to do is change this paradigm. And the approach we have taken is to think about microscopy in a new format. So in my lab, we invented what is called a chemical microscope. Instead of looking at the color of objects, we're actually going to look at the chemical composition of objects. So at every pixel now, we're going to measure the chemistry of different compounds that's present at that pixel in an image. So our data looks something like this. Instead of the flat 2-dimensional image, we have what we call a 3-dimensional data set. And at every pixel, we have, as a function of frequency of light, we have an optical signal. So just using light, we are able to sense the chemistry that's present in our sample. Now, this is a very complex signal, and a human eye cannot distinguish the very subtle differences between cancer and normal cells. So we actually use artificial intelligence to do that. And instead of the image like this, which has been present for 130 years, we actually seek to present the clinician with an image that looks like this. So now it's color-coded. Everywhere you see green are normal cells, are benign cells. Everywhere you see red is invasive breast cancer cells. And everything else, for example, that is present in the tissue can also be color-coded. and we can actually get much more information than we ever could previously. So now, instead of worrying about staining the tissue and doing microscopy, we actually can do this very quickly. But of course, we're not going to divorce ourselves from practice in the clinic. So actually, we can use artificial intelligence to not only get a picture like this, but we can actually get a stainless image without actually staining the tissue. So what we're hoping to do is change the process. Instead of the upper arrow being a multi-step process, we're actually just going to do chemical imaging and use AI to come at a diagnosis. Now you can imagine this opens up the whole diagnostic space. We can deploy this instrument anywhere. If we miniaturize it, we can put it even inside the body so that tomorrow we'll be able to diagnose cancer inside the body and not outside the body. So let me give you a few examples of how this works. So here's an example of a biopsy. So once the patient undergoes a mammogram, if there's something suspicious on the mammogram, a biopsy is extracted, and oftentimes they're sent home. And a few weeks later, they get a result. So our vision is, as soon as the tissue comes out of the body, it's put on our instrument, and within 30 minutes, we can tell you if there's cancer or not. The second example I'll share with you is once there's surgery and the patient is under surgery, when the tumor is taken out of the body, they want to make sure that every tumor cell was actually taken out of the body and nothing else was left. So on this side, on the left, you see an image. This is a red-green color-coded image. The red is the cancer, green is the normal, and you can see in this particular case in surgery, all the cancer was safely taken out and the tissue left inside is completely normal. So this image is generated completely from chemical image. The image on the right is what pathologists are used to seeing for 130 years, but this is also generated from chemical imaging and without any dyes and without any stains. All of this is accomplished in 20 minutes. So the patient is still being operated on, their tissue comes into this facility, we quickly measure it in 20 minutes, we can tell the surgeon it's safe, the patient is now treated properly, and we can complete the surgery. Another thing that happens when people are diagnosed with cancer is the cancer moves to their lymph nodes. So it's very important to see if there are lymph nodes present. Here on the right, I'll draw your attention to the bottom right. This is a clinical image, and where you see brown color, that's where cancer cells are present in the lymph node. On the left is a normal lymph node. There are no cancer cells here. We can give you the exact same image. So these are images on the top are generated by our technology, where we can actually talk about the cytokeratin positive or cancer cells, and there's no cancer cells here. Again, we can do this in 10 minutes. Right now, it takes 48 hours in the clinic to do this. But these are things that we can still do in the clinic. What about things that we cannot do in the clinic today? So here's an example. So currently, this is an example from cardiac tissue. So we can take tissue in the heart, which might have amyloids, and they want to know what kind of amyloid it is. So currently, it's a 2 to 3 week diagnostic process. What we were able to show is we can get the same accuracy in about 20 minutes. Finally, I'll talk about this a little bit more. What we want to do is not just diagnose disease. We actually want to use this new technology to tell you what will happen to the patient based on the chemical composition of their tissue. Is it a slow-growing disease and we can treat it a little less aggressively, or do we need very, very aggressive treatment? That's the kind of question we want to answer. Finally, all of cancer biology pretty much today is in 2 dimensions. And by using these kind of technologies, we can actually generate 3-dimensional views of cancer so that we can get the whole picture that was invisible to us just a few months ago even. So let us take a little bit deeper dive into the technology. Here's a stain. This is a completely virtual stain. And actually, we've done this now for many molecular stains. So not only can you do this for the primary diagnostic kind of stain in the clinic, we can also do this for many specialized decisions that are made in the clinic. So on the left are clinical images. On the right are images that are coming from our laboratory. And now if you're sharp-eyed, you'll say, okay, they kind of look like each other, but the images coming from the laboratory are not as crisp. They're not as good looking, perhaps. So how does one make images that are of clinical standard using new technology? That's the challenge. And that's the second part of the story. So once we have discovered a new approach to think about pathology, then we have to bring in engineering to say, how do we make this useful and how do we make this actually perform at a level that we are excited about? So what we're going to discuss today is how do we make this perform at the highest possible level? Then how do we make it accessible? And finally, how do we make it cheap and usable? So I promise you, this is the last slide with equations, but in a nutshell, the idea is that we have modeled a microscope from the source to the detector, and now we can optimize it. So just to tell you the impact, on the left-hand side are commercial images. So these are from a commercial microscope. On the right-hand side, based on the theory we developed in the laboratory, these are examples of microscopy image from my lab. So now we understand the limits of performance that we have to target. And now we're going to build instruments that actually perform at that limit. Once again, this is not a short period of time. We have to invent something, refine it, go back, improve it, and go on. So the lesson I want to leave with you here today, that over the last 10 or 15 years, we've actually systematically developed this technology where just a few years ago, it actually reached the top level of performance that it possibly can. So now that we've reached the top level of performance, we said, how can we use this technology? So we built a microscope that looks something like this. So this is actually existing in our lab at Illinois. And then we collaborated with our colleagues at Mayo Clinic. So here we're looking at biopsies. I showed this example previously. And what we're able to do here is find the normal regions and find those regions with cancer and make sure these little red regions that are there, cancer is present. We can quickly verify that and get the patient properly treated. So the idea, remember, was after mammography, in the 30 minutes that the patient is getting dressed, we'll be able to tell them whether their biopsy was normal or whether they have some cause for concern. Unfortunately, when we did this very high quality imaging, this actually took about 8 hours of data to acquire. So it was performing the right way, but not in the time that we actually wanted it to perform. So what do we do? Well, go back to the engineering. We went back and we redesigned the lenses. So these are lenses that nobody had thought about for about 80 years to see if we could make them better. It turns out we took up that challenge and we decided to model them and make them better. Then we actually physically built a whole microscope. This is all work done by University of Illinois students like yourself, right here in the lab. We actually physically designed this, built it, built all the electronics for it, integrated the whole system together, and then put in a layer of software. So this is a system that autonomously, you just load your sample and press a button, autonomously will go and scan the sample, make a decision, and give you the result. So completely automating this practice of pathology, which has been a very manual, involved process for 130 years. And the results look something like this. So this is a microscope. We have made 5 of these microscopes. There's 4 at the University of Illinois campus, and there's 1 actually at the Mayo Clinic Pathology Lab. So I'll share with you some results in the next few slides that we got from Mayo Clinic. But first, a word about the technological improvement. So on the left-hand side is the state-of-the-art instrument that is commercially available. On the right-hand side is our instrument. So we can get much sharper images and theoretically optimal images from our system. On the left here is a single field of view, so we can only see a little bit of tissue in a commercial instrument. In our instrument, we can see a lot more of the tissue itself. So by using this, we actually created a system where we could get very high quality images. Here's an image of the pathology in colon tissue, so colon cancer here. And we were able to acquire this image in about 20 minutes, as opposed to the 8 hours that you saw in the previous example. So we took this instrument, it's now sitting in the Mayo Clinic lab. So a patient is being operated upon, the tissue comes to the lab. They take one part of the tissue, give it to us. They take a second part of the tissue and go to Mayo Clinic in their standard processes. And then what we do is we present results like this. Here is an image of the surgical section. So these are tissues from surgery. And we've been able to tell you, for example, the tumor is here in this red line. We can tell the pathologist in 10 minutes, here's the tumor. They can zoom in into this box, for example. We've looked at here, all the red is cancer, all the green is normal tissue. In this case, there's some cancer here, which we can also pinpoint. And then without using any dyes, without using any stains, we can actually show them images that look like this. This is extracted from just this little part. So you can see these are cancer cells. Out here are normal cells, and you can also see that over here. In this particular case, this is completely normal tissue, and you can see the nice normal structure of the tissue. This is cancerous, and you can see that's cancer. So instead of the 30-minute process, intraoperatively that results in large amount of time being spent in surgery, we can cut down that time, cut down the effort, make surgeries faster, safer, and actually much more accurate than we could do manually. So we hope this technology gets more widespread use and can actually make a difference in more places. We've, of course, shown this now for many, many different organs. And actually, in all cases, the virtual staining that we do can actually be seen to be much higher resolution than even the real staining. So one day we imagine that a lower cost, better technology that offers you better views of cancer can actually help make better decisions that improve lives for people. So our imagination is this, that you will see tomorrow's pathology being this highly multicolored one. Wherever the eye of the clinician goes, it'll put a label there and tell you what that piece of tissue is. So that's great. This technology is on its way. It's moving towards the clinic. It's going to be used hopefully soon. So let's move to a second area, take a second deep dive. And the question here is, can technology provide a way to go beyond our current understanding with the idea that we can improve therapy decisions compared to what we do now? So our vision is the following, which is a really audacious vision. The idea here is, can we obtain a few cancer cells from a patient, take those cells, grow them in the lab, and make hundreds of tumors in the lab? Then we test combinations of drugs, or maybe even one drug, in the lab, and we treat the patient once very effectively and cure their cancer once and for all. So that's the idea. But the challenge is, how do you make hundreds of tumors that look the same in the lab from a small amount of cells? The second problem when you do that is, what about the complexity of human cancer? So actually, this is a large study, but essentially what we were able to show that it's not just the tumor cells, but it's actually the cells around the tumor that can tell us whether there's a high risk or a low risk cancer there. So it's very important not just to have the cancer cells in a mass, but also to have them organized along with other cells that are present in a tissue. So how does one do this? There's no real way to do it. So we took upon this as an engineering challenge. And several years ago, we said, okay, we can make these little toy systems. We can take tumor cells and we can put other cells, in this way. But, our bodies are not Petri dishes. So we don't have this kind of organization in our body. In fact, cancers are formed along ducts in our body, very complex structures. So what we decided to do was build a 3D printer. So here's a printer whose job it is to build the very fine biological structures we see in our bodies in nature. And the printer works like this, so you can see it in action. This is sped up by about a factor of 2, but you can see how the printer is actually printing very fine objects. And what it is out of the nozzle is coming out molten sugar that is solidifying as soon as we print it. And we can make exquisite things by engineering this printer. So you can see the Eiffel Tower there. We've printed a little model of a heart mesh, and you can see all those examples. But for our purposes, we built models like this. So this is meant to mimic breast cancer. There's a duct here, divides into 2, divides into 4, and so on. So we would print this out of sugar. We would coat it with a polymer, put some cancer cells on it, and organize the cells around the tumor to look like this. And then we'd withdraw the sugar, as you can see on the right. So the sugar is gone, which is the darker colored compound, and you're left with a cancer growing in a little test tube within its natural environment. So this was great. This was a technological breakthrough, but the problem was it wasn't fast enough. So now what we have to deal with is the speed and consistency that trades off against the complexity. So we focused here on understanding the limits of our complexity. And so we decided, let's focus on understanding the limits of our speed and consistency. So we were very fortunate that there was a new federal agency that was devised a couple of years ago. And we proposed to them a new way to engineer cancer tumors in the lab. And they were very receptive to this idea. And we were one of the first projects to be funded out of this agency. And the idea here is actually rather simple. The idea is we will take a few cells from a patient, grow them in a special way with our process. We will use advanced imaging that we have access to here at Illinois. We will build a computer program that then controls the growth conditions. So we can actually control the cancers in a very controlled way. So as opposed to complexity, what we're trying to do here is really control many, many, many tumors that we can grow in the lab. And what we had to do were a few more advances. We had to design the new media in which these would grow. We had to design new algorithms to determine how to control the growth. So for example, here, to my knowledge, is the first example where you can just tweak in a known way the exact same cells and either make them grow faster, than the tumor in the human body, which is the blue curve here, or make them grow at the same pace, which is the orange curve here, or actually make them grow even slower in a controlled way, which is the yellow curve here. So now we are able to do experimentation in a high throughput way, completely automated, by using advanced technology that tells us if we can monitor the tumor health or normal tissue health. And again, this is the chemical imaging that I was telling you about a few slides ago. So when we put all these three things together, lo and behold, we've been able to grow tumors now in a very controlled way in the laboratory. And actually, we've been able to grow more than 15,000 tumors, all accurate within 10% of each other. And this very large scale growth now allows us to take a few cells, grow them into these large numbers of tumors, and maybe test at the individual patient what we can do. So this technology, we hope in a few years, will be moved towards actually use, and will obviously change the way we think about therapy as well. In the last 5 minutes or so here, let me share with you the third deep dive. I kind of alluded to this a little bit in the past. And the idea here is bringing a cancer cell to life in a computer. The reason we're trying to do this is cancer is such a complex disease to study. If we can make computer models of how cancer initiates and progresses, then we have a shot at truly understanding the disease and maybe intervening in the right way in this disease. So the vision here is to record the position and identity of every molecule in a cell, make that list available to our colleagues in computer science and biophysics, have them make a computer model, and then run the model under different scenarios by stressing it with oxygen, with nutrients, with things like compounds in cigarette smoke, et cetera, and see how we actually can see cancers move. So I mentioned this is a very large effort between our university, which is leading this effort, and many other people across the world. We think it's a really new way to do biology inside a computer, and do it in a very quantitative, understandable way that can allow us predictive rather than observational biology in the future. So we call our center the Center for Quantitative Cell Biology, and it's a really new way to think about it. But at the heart of this problem is a challenge. The challenge is, how do we record each and every molecule in a complex mixture such as a cell? And so there's technology that was invented not at Illinois, somewhere else, but when we got our hands on this, it actually had a number of limitations. So as you might imagine, the Illinois way of handling this challenge is actually to go back to fundamentals. So we spent some time looking at the fundamentals of how images are formed with this technology, and we could actually see that there were some gaps that the field didn't quite understand. But by a proper theoretical modeling, we were actually able to devise a whole new way to make these measurements. And so this is one of the inventions from Illinois, and it provides us the ability to actually record very, very small domains from a cell. So this is, I'd like to share with you the first chemical image of a cell at 40 nanometer resolution. So we have never seen as a species, as a human being, ever cells with this resolution, with this kind of chemical contrast. So we got these images. For example, you can see the cytoskeleton of the cell here, individual filaments. We think these little dots here are actually individual protein complexes, individual subcellular organelles to that extent that we can actually measure. So we're cataloging these as we speak, but incredible complexity of the cell that's now being revealed because we have better technology. And we're going to build up our information from here to actually eventually use this in a model system. So this is all good and great, but one of the most fundamental things and fun things about our campus is that we're able to take these advances and move them from everybody can get involved, from a general middle school student or maybe even elementary school students all the way to very high-end scientists. So I'll give you a few examples of how we're doing that, and it's really one of the fun parts of the work we do here on campus. So as I showed you a little while ago, we can now take chemical images of cells and tissues. So here's a chemical image of a cell. And what we're doing here is color coding different kinds of components of a cell with different colors. And then we're importing it into Minecraft. So here's a Minecraft model of a cell. We call it a craft cell. And essentially, your middle school student can actually go out to our website of the Center for Quantitative Cell Biology and download the software that actually allows them to take cells and models into Minecraft, and they can play with them all they would like. So we're going to disseminate more of these models. We're going to have much more exciting data. If you want to get involved in this, it's very easy now to go forward with this. We're also pioneering a whole new way of educating students. So, as I mentioned previously, we're pioneering this idea of cancer engineering, the idea of bringing fundamental engineering innovations and engineering thinking to oncology. And we've made investments across the board. So here's some of our wonderful students who are sponsored by the Cancer Center at the graduate level through a training grant at the federal level. We've also devised a wonderful undergraduate program. If you haven't had a chance to check this out, I would put in Cancer Scholars Program at UIUC and go search for it. We also have tremendous opportunities. If you know a bright high school student, we have a summer program that they can apply to and come to on campus and look at it. We're very fortunate that we're able to do these things here on our campus, try out these new educational models, because the country is looking at us for these solutions. And actually, we were able to organize a National Academies meeting last year. There's a report if you're interested to go look at it. Here's the report on cancer engineering and cancer engineering education as we go forward. So we think we're at the cusp of something very interesting. We're at the leading edge of this movement around bringing engineering and basic science to cancer to fundamentally change things. And I think we have to be very proud of our university having established this pipeline all the way from fundamental discoveries to building practical technologies to taking it all to use. And of course, this is a team sport, as I mentioned many times. So I want to start by thanking those in my lab who contributed to the work that you saw today. There's a whole bunch of great graduate students, postdoctoral fellows, scientists, administrators, and staff, as well as undergraduate students who contribute to this. There's a number of collaborators, and of course, we're very, very grateful for the federal funding that we obtained from NIH, from National Science Foundation, from our colleagues at Mayo Clinic, and agencies like ARPA-H. With that, I just want to reiterate our message for today. We're really all about in the Cancer Center from discovery to use. We're all about providing practical cancer solutions and really improving lives. And if you want to get more engaged with us or involved in cancer research on campus, please feel free to email us at the email address down there at the bottom. Thank you for your attention, and I'm happy to take questions.
Thank you so much, Rohit. That was a really inspiring and exciting presentation. We've got quite a few questions coming in here. So let's start with, does the center currently leverage any collaborations with the biopharmaceutical industry?
Yeah, that's a good question, Ellen. So yes, we do. And at the right time, we want to leverage the right partner to help us do that. For example, what we are trying to do on campus is develop new molecules as therapeutics. Well, we want to make sure that we have enough evidence so that when we engage a partner, they're very confident that this is going to go forward. It'll speed up their efforts. It'll speed up the impact of our discovery. So we want to do this in a very thoughtful, individual way for each invention that happens on campus.
That kind of leads us into this other question here. What are the challenges in moving drugs invented at Illinois into patients, and how soon can we expect these drugs to be available?
The number one challenge is organizing the effort. Doing a test requires, thankfully, a lot of safety, a lot of testing, a lot of efficacy, and investment over a period of time. And of course, as everyone knows today, it's a challenging funding environment at the federal level. Industry is increasingly focused on very short-term results. They're concerned about the next quarter. So we are trying very hard to figure out how we can garner support to keep these long-term efforts going. If we want to start on proving a new drug is useful, once we've done the science on campus, it often takes 2 to 3, 4 years to prove that. In the meantime, we need the sustained support. So actually, we've been very fortunate that we have some people in our community, some of our alums, in fact, who are supporting these efforts. So if there's anyone on the call who wants to get involved and help support these efforts, please contact us. We'll be very grateful. And of course, we welcome any kind of support to move that forward. But the number one problem today is just the support we have for sustained activity to move forward. We know what to do, just cannot do it.
In terms of support and collaboration, as you mentioned, could you just mention a few things that you've done in collaboration with the Carle Illinois Medical School?
Yeah, we actually think of the Cancer Center as a global collaborator. So obviously, Carl Illinois College of Medicine is our favorite, collaborator. They're local. They're our own college, and so on. But there is no college, there is no cancer center in the world that can address every single need in the cancer world. So what we've done is obviously established relationships with Carl Illinois College of Medicine. We have helped recruit their faculty who are cancer center members and also members in the Carl Illinois College of Medicine. We work with Carl physicians. We also work with OSF physicians locally in a very strong partnership. We've also formed partnerships with Mayo Clinic, with Washington University St. Louis, with the University of Arizona, with Memorial Sloan Kettering. And the wonderful part is all of them recognize that the University of Illinois has something to offer for their patients and their clinicians.
Mhm.
This large cohort of basic science, and I really apologize, I was only able to cover a few examples. I could have given a 4 or 5 hour talk on all the various innovations, and then we wouldn't be done even, right? So there's a huge amount of basic science. And I think the success of the Cancer Center is partnering with the right partner on the clinical realm who has the right number of clinicians, the patient volumes, the patient population that we can help. And we're about helping everyone in the world as soon as we can.
How can CCIL research bring down the cost of cancer care?
That's a great question. This is something that really occupies us, day and night. So the central premise is that when we discover new ways of doing things, most often with technology, we have to find new ways of doing what we had done in a complex, convoluted way in the past. So I'll give you one example. For example, chemotherapy today might take 8 weeks of once a week sessions where one has to drive from where they're living to a facility that can offer them chemotherapy, stay there for a few hours, often take somebody along with them. So it's a couple of people at least driving at a certain distance over multiple weeks to get therapy that's kind of not very specific. It has a lot of side effects. It has other complications. Our researchers are thinking of ways in which we can make a single pill, so no chemotherapy, a single pill. If you have a kind of breast cancer, you take this pill and hopefully the cancer is gone forever. So by using these very powerful ways to devise medicines, we're actually changing how you're going to deliver cancer care. And I guarantee you, providing a single pill that's invented at the University of Illinois will have lower cost associated with cancer care than 8 rounds of chemo, where you're not only going to the place and the facilities and so on that maintain the chemotherapy, the chemotherapeutics are very expensive, and you're taking off of work to go 8 times to this place. You're not feeling so good. You're taking a colleague who's also taking off work and so on. So it's not just the cost of healthcare, it's the cost to the human being that hopefully we can reduce by a new way to think about therapy, about diagnosis, and so on that you saw here.
Yeah, absolutely. Um, this one's kind of interesting. Basic research often yields unexpected results. Do you have any examples of failed research that has opened up new avenues of investigation?
Well, let me. You saw a failed experiment today. We tried to build this very complex 3D printer, and it didn't quite go the way we thought. Right? We thought we could print out, objects. So we had to take a step back, re-engineer, refocus. Right? We try to solve a problem, which is, can we make enough tumors in the lab to be able to do the science and the experimentation that is required, and then over time build complexity. So the way we look at it in the Cancer Center is no failure is permanent, but we can certainly learn from it, make progress that is useful in the interim, and then come back to make really more ambitious, advances that perhaps their time is not right now, and maybe in a few years we can come back to it.
I love that. Let's see, we've got time for a couple more. When will tumor models of individual patients become a reality?
Oh, that not fast enough, right? I wish we could have this technology tomorrow. We've been really working hard for years, and so have many, many people across the country. Let me tell you that I have never seen the consistency and the volume and the reproducibility that I just showed you on one of the slides anywhere in the world. So I think we are about as far ahead as one could be. The next step is we are actually that was phase one of our project. We just got awarded phase two. So in the next step, we're going to take tumors that were implanted in mice and make sure we can reproduce those. And the third step would be to actually test on human samples. So if everything goes well, and again, this idea of failing, we anticipate that we might fail a little bit. But I hope if we progress around the roadmap that we have laid out with the technological approach that we've taken, it might be 3 to 5 years where we're able to test on humans and make sure we're able to make human tumors in the lab. And then, there's a little bit of process from there to actually moving it into the clinic. But the technological sort of breakthrough or the gap that we need to solve, I believe we can solve in the next 3 years. But provided we maintain continued support for this project, and we can put in the effort that is required over the next 3 years.
How do you think that computer models of cancer will be used?
We think they will be used at multiple scales. So imagine a computer model of a whole human. So it takes in the data from your wearables, from your cell phone usage, from your environment sensing. Was there a wildfire 20 years ago in your area when you were present there, right? So at one level, it's a what we call very macroscopic model. And at the other extreme is the cell model that I showed you. So imagine every molecule in a cell and every cell in a tumor with millions of cancer cells working together and understanding this thing. So the point is that there will be a whole range of tumors that will be simulated, a whole range of cancers that will be in silico inside a computer. And of course, the University of Illinois is the premier place to do computing in the world, right? So it's only fair that all this movement start here. But my vision or my anticipation is that we will have models that are maybe suited to predictive at the human level. We have models that are suited to the understanding and the scientific, questions at the heart of cancer. And we'll be able to we'll be able to do research faster through some models. We'll be able to predict risk for humans at some model. We'll be using another set of models to predict, how a particular human might respond to a particular drug that is administered at a particular dosage on a particular day. And using those scenarios, we'll be able to fine tune therapy. The closest I can tell you is that I'm not sure, exactly when or how, but let me be brave and predict that one day these models will be in cancer care the way AI is in everything that we do today in our lives, right? In your phone and booking a ticket to go somewhere, and pretty much anything, even searching for information online. AI is so integrated into our lives. As computing and storage expands exponentially, as it is doing now, you'll see more complex versions of AI, which include models of cancer. So maybe in 5 to 10 years, you will actually start seeing models in patients' hands where they can ask what if questions. They can say, well, I have two ways of thinking about therapy. Do I go for this one or do I go for that one? Please run a simulation to show me what will happen. Right. So I anticipate those models will be perhaps one of the most impactful things coming forth in a few years.
Yeah, that's very inspiring and exciting to consider. Um, why is the microenvironment of cancers not currently used for clinical diagnostics?
Because it is so difficult to keep many pieces of information in the human mind. So all the cancer diagnostic criteria we have today are based on a human looking through a microscope and making decisions. And humans can only keep maybe 3 or 4 things straight. That's why, for example, when we go to the grocery store, we make a list, because by the time we think about the first 3 things, we forget about the fourth thing, right? So in most humans are limited to just a few things that we can communicate and collaboratively keep in mind. The second thing is all diagnostic criteria have to be sort of universally agreed, right? Like a whole range of clinicians have to say, we're diagnosing cancer the exact same way. And to communicate the complexity of that, you can't make it too complex. So you can say, oh, grade 1 cancer, grade 2, grade 3, grade 4. You never say grade 75 of cancer because it's too complex. So generally 5 things or fewer. So what happens is we're able to describe cancer cells in 5 descriptors, but we can't describe what's happening around the cancer cell. So imagine what really happens. What really happens in cancer is the cancer is growing, the body is sending defense mechanisms like fibroblast cells, like immune cells. There's blood vessels growing to supply nutrients to the growing cancer. Now, if all of these things are present, only then does the cancer grow. So by measuring the microenvironment, we actually have a shot at determining which cancers are dangerous and which are not, for example, because if there's no blood supply, well, then the cancer is not going to grow. But it's too hard for a human to keep these pieces of information in their brain together. So this computer treatment of biopsies, computer analysis, is really a tremendous technological advance because we can now use the microenvironment information to add to the cancer cell information that we've already got. So I definitely, you're already seeing things in practice that are being used in many clinical centers across the country. But I think as time goes on, you'll start to see more and more complex information being used to predict how cancer is going to behave. And then, of course, that will make us much more precise in determining exactly what kind of cancer there is, so we can treat it better with less side effects and so on.
Yeah, absolutely. I think we have time probably for one more. So I'll just ask, how is the Cancer Center at Illinois different from other centers?
We are the only cancer center in the nation that is focusing on bringing engineering from the ground up into cancer practice. And there's a lot of places which might take things that are engineered, then bring them into practice. Certainly, that's a very positive thing. There are places that might do a little bit of technology and inspire that. The University of Illinois is one of the largest collection of engineering, physical scientists, veterinary scientists, and basic scientists anywhere in the United States. Maybe more than 700 faculty members who can potentially contribute to cancer research. So what we have decided to do is really turn our attention without worrying about what happens to a patient tomorrow. We want to turn our attention into what are possible solutions that are long lasting. And we want to bring in innovation right at the ground floor so that we always keep an eye on, is it going to be low cost? Is it going to be widely deployable? If you bring in engineering, if you bring in development too late in the process, it's already optimized for something else. So many technologies that are used in cancer are high cost because we did not worry about cost at the initial stage. We just sort of invented and tried to solve the problem. So by bringing our fields together the way our cancer center has, this relentless focus on improving things while lowering costs, while improving accessibility to people, that's what makes us unique. In addition to our focus on long-term solutions as opposed to short-term fixes. And there's, to my knowledge, no other cancer center that's doing it at our scale with our dedication.
Thank you very much for everything tonight, Dr. Bhargava. Um, Someone did ask if the presentation slides will be made available. We will share them in a follow-up email following this to the attendees who attended. I also want to say, as a reminder, a recording of tonight's presentation will be available on our website within the next 10 business days. Thanks everybody for joining us tonight. Have a great evening, and ILL.
And I thank you, Ellen.